Chapter 12 is about the nervous system. And one of the things to consider is how do you categorize, divide, and make sense of it? So one of the ways to do that is anatomical divisions. CNS stands for central nervous system. That's your brain and spinal cord. PNS stands for peripheral nervous system. That's all the other nerves that branch away from your brain and spinal cord. The peripheral nervous system is divided up into two major parts, the sensory part and the motor part. The sensory part has to do with taking in information, whereas the motor part has to do with controlling muscles and glands. ANS stands for autonomic nervous system. You don't control that, and it's broken down into two parts. The sympathetic nervous system, which is your flight or fight response, and the parasympathetic nervous system, which is your rest and digest response. It's like getting ready for action and then recovering afterwards. Somatic refers to body senses. So those would be things like your eyes, ears, nose, things that are taking in and collecting information. Another way to divide the nervous system is functionally. In other words, how does it work? One way to categorize that is the sensory function, where you're bringing information in from your external environment, and then the motor function, moving muscles or glands to respond to that. And in between those two is the integrative function. That's where your brain and spinal cord come in, making sense of that information and deciding what to do next. Sometimes the integration is called association, and I prefer the association because it makes this acronym SAM. Sensory coming in, association figures out what to do with it, motor is the result. And we can classify neurons that way. Neurons are nerve cells. Remember all the way back to nervous tissue, neurons and neuroglial cells. The neurons, that's the workhorses of the nervous system, they can be categorized in those three ways. Another functional division has to do with how does it control the body. So in this case, the somatic nervous system is going to be your conscious perception of things, your voluntary responses, your reflexes. The autonomic nervous system is going to control things like smooth and cardiac muscle. It'll control glands, things that you don't have direct control over. And the enteric has to do with the smooth muscles and glands of your digestive tract. So again, it has to do more with the functional control. Here's a neuron. It's the workhorse of the nervous system. A is pointing to the cell body. B is an axon. C is going to be a glial cell, an oligodendrite, and it's working to make this myelin sheath. That would be E, the part of that myelin sheath. D are the gaps in the myelin sheath. Those are called nodes of Ranvier, these little spaces in between the way those cells roll up around the axon. F is a synapse. That's a space between the nerve and a muscle or gland. So whatever it's about to control that muscle or gland, there's a little gap. They don't actually connect. G are dendrites. Dendrites collect information and then send it down the axon. And then H is pointing to the cell membrane of that neuron. Here's a picture of a neuron, and you can see all the organelles. And the reason I like this picture is because it shows that cells are not just these round circles with all the organelles in the same proportions, but different organelles relate to different cells differently. In other words, a cell like this is going to be very dependent on things like the cytoskeleton in order for that axon to be very long. They're going to have a lot of mitochondria because mitochondria are needed to make ATP for active transport. It's the active transport of a nerve cell that's going to allow it to have an electrical impulse. The vesicles are going to be very important at the end of that nerve cell because that's how neurotransmitters, these chemicals, get released from the nerve cell to control that muscle or gland that it's associated with. So different organelles have different significance depending on the job of that cell in that tissue. Well, we have two major nerve cells in the nervous system, neurons and neuroglial cells. Sometimes they're just referred to as glial cells. Now, glial cells can multiply and divide very easily, whereas neurons don't do that so much. Tumors are just cancers where we have uncontrolled cell division. So glial cells are the ones that are typically dividing more rapidly. And in the case of a brain tumors, they're dividing uncontrollably. Myelin are the cells, like all the cells together. We call that a sheath, that myelin sheath that lines an axon. And that's going to speed up the nerve impulses. It's going to allow those impulses to travel much faster. And then little spaces between those Schwann cells that make up the myelin sheath are called the nodes of Ranvier. So there's little gaps in that sheath. Here are three different kinds of neurons. You'll notice that it has to do with the appendage that juts out of the cell. So if there's one appendage, unipolar. If there's two, bipolar. If there's a lot, multipolar. Typically, for diagrams, we see multipolar neurons. Neuroglial cells are the supportive cells. And there's a bunch of different neuroglial cells, and the way that they support cells are different. So astrocytes, they look like stars or star shape, and they're going to support mostly the central nervous system. Whereas oligodendrites, those are the ones connected to the axons, they're going to form the myelin. The microglia, they fight diseases, and the epidermal cells are going to filter blood and make cerebral spinal fluid. So each of these glial cells, different roles, but they all support the neuron. How does a nerve impulse get transmitted? It starts with resting potential. Resting potential means we have a positive charge, a significant positive charge on the outside of the axon. And we have a significant negative charge on the inside of the axon. That difference in charge is going to create our electricity, that impulse that's traveling along the axon. Sodium ions, they're positively charged. They're in very high concentration on the outside of the neuron. Potassium are also positive, and they're in high concentration on the inside. You might be wondering then, if they're both positive, how do we have this charge difference? Well, that's because inside the nerve cell, we have large polyatomic ions that are negatively charged. And right now, they're overpowering the charge of the potassium, so it's overall negative. When we reach a threshold, that is, when the stimulus reaches a certain threshold, the channels will open, sodium will rush in, and potassium will begin to leak out. That starts a cascading event where along the axon, those channels are opening, sodium is rushing in because it's going from this high concentration to a low concentration. Potassium is leaking out because it's going from a high concentration to a low concentration. So you see the positives rushing in, and then as those potassiums leak out, you can see it's like the negatives leaking out, like changing that charge on either side of that axon. Think of it kind of like dominoes. Touching the domino and not tipping it over, that's the threshold. I've got to touch it hard enough so that it begins to fall. But once it falls, it's going to start this cascade event. When it starts this event and the domino falls, it will fall with the same force the whole length of my domino setup, or the whole length of the axon. From one segment to the next, those channels are opening and that impulse is falling. So that's the idea, that it does not diminish as it moves along the axon. The force of that action potential doesn't diminish. Once the dominoes have fallen over, can I knock them over again? And the answer is no. I have to set them up before I can knock them down again. In a similar way, the action potential needs to be reset. That resting potential needs to be reset before we can have another action potential. Take a look at these three nerves. What does the action potential travels down the axon? You can see that in the first one is traveling the slowest, in the second one is much faster, and the third one is faster still. That's the difference between myelinated nerves and unmyelinated ones. Myelinated nerves travel very quickly, the impulses. And it's because they're jumping from one node of Ranvier to the next. Thicker nerves have faster impulses than thinner ones. This is a difference between things like your knee-jerk reflex and your eyes adjusting in dim light. Your knee-jerk reflex is myelinated nerves. Your eyes adjusting in dim light, that's going to be an unmyelinated nerve. Saltatory conduction references that jumping from one node of Ranvier to the next. So a thick myelinated nerve is going to be the fastest in terms of sending impulses. Think of it like running a race where you have two contenders. Person A has to put both feet in every block of the sidewalk. Person B can kind of spring from one block over a block to the next one. By skipping those blocks, they can run a lot faster than the person who has to put their feet in every single one. That's the advantage of myelin. You're skipping from one node of Ranvier to the next. This is what it looks like graphically. The resting potential is negative, so that's why we start at the negative 70. If the threshold reaches to the negative 55, the stimulus, the light, the heat, the touch, the sound, it's got to reach that threshold. Once it does, there's going to be a nerve impulse. Just like once you tip over the domino, there's no going back. It's going to knock them all over. That's the idea of all or none, where once the domino starts to fall, it falls all the way. Once the neuron starts to depolarize, it depolarizes all the way. So it's the same process every single time. As soon as you hit that threshold, it's going up to positive 30, and then the potassium is going to start to leak out. That's the repolarization part. And then we have this refractory period where we can't knock over the dominoes until they reset again. That resetting is the sodium-potassium pump, pumping the sodium out of the nerve cell against the concentration gradient, pumping the potassium into the nerve cell. It's going to cost you energy. That's why the mitochondria are so important. In order to get that difference in charge so that we can have another action potential. All right, so that's the importance of that sodium-potassium pump. That's the refractory period, the time it takes to repolarize and get everything back to normal. All right, so watch the animation again as the sodium is rushing in, the potassium is leaking out, and then it takes a little time to push that sodium back out. That's the sodium-potassium pump. That little gap is called the refractory period, resetting that domino, getting it to stand back up. But that's how a nerve impulse is irreversible. In other words, because this section is in that refractory period, when this section depolarizes, it can't stimulate that section to open those gates again because they're working that sodium-potassium pump. So the nerve impulse only travels in one direction. It can't travel backwards. That's the idea of irreversible. And it doesn't diminish as it goes. That's the idea of non-decremental. Like it doesn't get slower and slower. It's like dominoes. Once they fall with the same force, the last one as the first one. What happens when the nerve impulse gets to the end? So when it gets to the end, it's releasing neurotransmitters. Those are the little vesicles. Those neurotransmitters are going to cross the gap between the neuron and the next thing. Sometimes the next thing is a nerve, sometimes a muscle, sometimes a gland. But those receptors will collect that neurotransmitter, and that's going to stimulate it to either contract or release a hormone or send that nerve impulse farther along. You'll notice those neurotransmitters are recollected then. They're reused. But it's got to cross that gap. So what if a nerve is connected to a lot of other nerves and it's getting mixed signals from neurotransmitters? Some are excitatory, yes, 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 yes. Some are inhibitory, no, 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 no. So you can see like the green ones are excitatory, the red ones are inhibitory, no, 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 no. So what happens? It's very democratic. In this case, if it gets more excitatory impulses, then yes is the answer. If it gets less, then no is the answer. Here's an anatomy student studying away, and they have this impulse for milk and cookies. But no, I've got to focus on my studying. No, I really want milk and cookies. So what's it going to be? Yes, 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 or no, no, no? Well, I think we know the answer to that one. Neurotransmitters have different effects depending on where they're located. If the neurotransmitter is in the central nervous system, it might have one effect and a slightly different effect if it's part of the peripheral nervous system. Look at acetylcholine. In the central nervous system has a lot to do with memory and is associated with Alzheimer's. In the peripheral nervous system, it has to do with muscle contraction. Dopamine has a lot to do with pleasure sensation and addiction. Low levels of dopamine are sometimes associated with Parkinson's disease, the shaking disease. In the peripheral nervous system, it has to do with skeletal muscle tone. So different neurotransmitters, sometimes different depending on whether they're part of the central nervous system or the peripheral nervous system. Well, that's a quick, short summary of Chapter 12, the nervous system.